Recording in progress.
Yes. So, is there any doubt in the book? Please go through. This is the last class. You can use it for doubt clarification. Please go through the book, the yellow book. If anything which you cannot follow, please ask me. And because you're going to have your test, so here in the center, for all of the chapters which we finished there. Heredity இருக்கும், how the organism reproduce. That's அதில் please be sure with the reproduction in flowering plants. Bookல இருக்கிறது மட்டும் இல்லாம it just focus well on this particular thing also. And you saw the questions, like some questions may be new to you, but still if you cannot able to answer some questions, don't worry, and you learn the question, you know the questions, and then we will discuss about the answers. Okay, so just check your book. If anything which you cannot follow, we can use it. And hereafter, we don't give any time for revision for this particular chapter. So go through. And Rohit, for you, the checklist. So what and all you should know for everybody. What is important in this particular chapter? The terms, all the terms. Many terms are there. I think I dictated how many terms to you? Did I dictate any terms? 20 to 25 terms you should know at least. Okay. But you should know, if I would have told here and there, like genes, alleles, multiple alleles. Okay. You know these terms? Genes, at least note down now, important terms. You have to find out the answer for that. Genes, alleles, multiple alleles. Okay. And then genetics, heredity, variation, locus, dominance, recessiveness, homozygous, heterozygous, genotype, phenotype, yeah, are you all following this? Writing, noting down these terms. Write it. Which one? Dominance, recessiveness, hmm? Where? Page number 27. Which one? Terms one. They have dissimilar alleles for a specific. They produce two types of gametes. I'll tell you. Let him copy. I will tell you. I told you already, but anyway, I will remind you. See, finished up. Filial generation, progeny. Or diploid. See, primordial germ cell has X, Y chromosome in a diploid cell. This is 2N in a diploid cell. By meiosis, what occurs? They undergo duplication process. So X becomes XX duplicated. Y becomes Y duplicated. So logically 2N becomes 4N. right? And this 4n divided into 2n, um, 2n, 2n means either you can say xx or they are separated y y, okay, and then this separated into sperm cell with x, another sperm cell with x, here sperm cell with y, this one sperm cell with y. So how many type of gametes? Heterozygous male produced, because male is heterozygous. How can you say it is male is heterozygous? XY, sex chromosomes are 2, male. So heterozygous. In meiosis, finally, one type of sperm cell with X, one type of sperm cell with Y chromosome. Are there two type of sperm cell? Yes. That's why data like heterogamete male produces two type of gametes, two type of sperm cells. Okay. Fine. That's it. And then what are the other terms? Here all the terms, different type of process is listed. You please get your book, okay, later after the class. So what are the thing? Mono, hybrid, different type of process. Mono, hybrid, cross, and then di, hybrid, cross, and then tri-hybrid cross also. Sometimes they may ask. Okay, monohybrid cross, dihybrid cross, and then test cross. Then back cross. No down. Monohybrid cross, dihybrid cross, trihybrid cross, test cross, back cross, reciprocal cross. Three pairs of alleles. Okay, cross one parent with three set of alleles. So two parents with one three character. For example, Mendel's hand. Dihybrid cross, what you included? Seed order, size is, sorry, shape is round, color is yellow. Along with if you include stem height. Means no stem height, tall plant or dwarf plant. So three crosses. Okay, fine. That's the hybrid cross. Three pair of inheritance of three pairs of contrasting character. Of course, there are many, but still at least these terms. And then alternative form of a gene. Alleles are alternative form of a gene. The term refers to each of the member of genetic pair or alternative form. Is it clear? Hmm? Fine, good. Next. So you have to understand the terms, and then about Grah or Mendel means they will ask why he is successful, why Mendel was successful, successful in his experiments. They will ask, and then why Mendel selected pea plant. Okay, that they will ask, and then what are what are the 7 contrasting traits selected by Mendel. Okay, what are the contrasting traits selected by Mendel? Are you noting down? Please note down the questions. What? Why Mendel was successful in his experiment? What you are supposed to write? See, he introduced statistics and mathematics in science. Okay, he carried out large number of experiments. Proper documentation and observation was done. He documented everything. He observed, proper observation and documentation was done. Okay, just a minute. I'm getting one. எப்படி இருக்காங்க? சரி, நீங்க எனக்கு கரெக்டா 5:30க்கு 6:30க்கு கால் பண்ணுங்க. 6:25 30 6:30. எனக்கு ரீசார்ஜ் இல்ல. Bye. Okay. Sorry. So proper documentation, proper observation, and the introduction of statistics and mathematics into science குள்ளார. இதெல்லாம் தான் important reason என்ன by which Mendel succeeded in his experiments. Okay, large sample size means 25,000 plants. Okay, and simple techniques. Okay, fine. And then why pea plant? Why pea plant? Well, contrasting characters, easy to cultivate, observable traits. Okay, you can observe the easy traits and all. Okay, 7 contrasting characters. And in tree plant, self-pollination called cross-pollination. Everything can be done. It's a bisexual flower. Yeah. Okay, bisexual. Yeah. So easily you can do any kind of pollination and short lifespan. Okay, in few weeks. First one now, Graham Mendel's experiment. Why Graham Mendel succeeded? That one. Okay. So introduced maths and mathematics அப்படின்னு எழுதுங்க. Stats in science. ஒன்று. Okay. And then his experiments involved large sample. Okay, large sample size, or you can say large number of samples. Okay, and then correct observation. Proper observation and interpretation of results. Okay, correct observation and interpretation of results. Okay, then copy down. Okay, and then in your book the points given can be noted. Selected, maintained. When selected with a maintained pure lines for all contrasting traits, or you can say characters. Okay, traits. Okay, and then all 7 traits he studied one at a time. All 7 characters those were studied one after other. Okay, so these are all the main reasons, okay, for Graham Mendel's success. Is it clear? Fine. And then the 7 characters also, you know. Next, they will be asking to work out monohybrid cross, dihybrid cross. You must know how to work out mono, hybrid, cross, and dihybrid cross. Okay, both you have to work out that part. And then definitely you should be able to explain laws of inheritance. No, don't hurry. Definitely you should know how to work out monohybrid cross and dihybrid cross. Okay. And then what are the laws of inheritance? Law of dominance. அடுத்து law of segregation, and then law of, what is the third law? Independent assortment. Okay, you should be able to define all Mendelian laws. Explain all Mendelian laws. Okay, explanation of all Mendelian laws. So these things, and apart from that, Kapida. Theories for understanding, sometimes they may ask, but very rare. In your class test or whatever may be asked, because they extract questions from past papers. So that time, these questions are there. So sometimes you get some of the questions related to that. Okay, fine. And then sex determination. Apart from that, what you should know? Sex determination in your book, right? Sex determination. Sex determination in humans. Sex determination in Drosophila. Sex determination in honeybees. Okay, different organisms, humans, Drosophila, and then birds. What factors contributes in the determination of sexes in different organisms? Okay, so that's the another important thing from this particular chapter. Drosophila, like a fruit fly. They call what? Fruit fly. Fruit flies, which is very, very small. நம்ம கிச்சன்ல இருக்கும் பனானா ஃப்ரூட்ஸ் பீல்லாம் போட்டா அதுல பழைய சாதம் போட்டா அதுல வந்து உட்கார்ற இந்த குட்டி 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 இன்செக்ட் லைக் திங் பறக்கும். இன்செக்ட் குட்டி ஈ மாதிரி முக்கியம் பாருங்க. பனானா எல்லாம் பார்க்கும்போது வந்து உட்காந்துருக்கும் பாருங்க, சின்ன சின்னதா, வெரி ஸ்மால். நார்மல் ஹவுஸ் லைஃப் கிடையாது. Housefly is bigger, but Drosophila is very smaller. குண்டு சீப்பின் இந்த ரொம்ப சின்னதா இருக்கும், ஒரு கடுகு சைஸ் தான் இருக்கும். கடுகு பார்த்திருக்கீங்களா? ஸோ அந்த சைஸ் தான் இருக்கும். ஓகே. And apart from that, you should be thorough with what? The terms, especially for board pattern. Sometimes they may ask many questions about what? Molecular genetics. What is molecular genetics? What's molecular genetics? Study of molecules, which determines inheritability. That is molecular genetics. All questions, genes, chromosomes, DNA, silent feature of DNA, all these things you should know. Okay. Fine. So go through what else based on your textbook as well as for this important. Why? பிரிக்காதீங்க. Definitely. That will be repeated here, what I said. Okay. You can refer that. Yes, Kaushik and Vijay Lakshmi, are you there online?
Yes, sir.
Yes. Kaushik, these are all the checklists. Like you must know. Okay.
Yes, sir, and that's
This all these things are the checklist. Vijay Lakshmi, can you hear me?
yes, sir.
Yeah, they are the checklist. You must know, right?
Okay, sir.
Fine, good. And please go through that yellow book material. If you people are having any doubt, you can ask me. So, and soon, I think today itself we will be finishing this chapter. And so you should be thorough with all the things. So if you cannot able to understand anything, you can post your questions.
Okay, sir.
For that only today is actually. Yeah, molecular structure of a gene. See, understand these terms. What are the terms explained? Molecular structure of a gene. Okay, what are the terms given here? Recon, muton, cistron, and then replicon. Okay, 4 important terms you should know. Recon is a small unit of DNA capable of crossing over and recombination. See, if this is the chromosome and they are the sister chromatids, this is one chromosome and this is another chromosome. One is paternal and one is maternal. So, recon means this portion. For example, if they are closer to each other, this portion, the smallest portion, can go for crossing over. Means here, by the end results, for example, in this arm, finally, one blue gene can come here. And then in another arm, because of genetic exchange, one red gene can go here. Can you understand? One little portion got exchanged during crossing over. This process is what? Crossing over. Okay? So, the simplest part where crossing over is called what? Recon. Okay? And what is muton? When this crossing over occurs, the damage can occur. Damage to the structure of a gene. Damage to the structure of gene occurs. That results in what? Mutation. So, in which part of the gene the damage occurred? That is called what? Muton. Okay? Damaged portion of a gene is called what? Muton. And then cistron. For example, active portion of a gene. This is the structure of cistron. DNA is double stranded. And one strand runs in 5' to 3' direction. Another strand runs in 5' to 3', like this. So, here one region is there called P region. Here one region is called what? T region. P stands for what? Promoter. T stands for what? Terminator. Okay? And the two strands, one strand is actually called what? Template strand. Template strand. Another strand is called what? This strand is called what? Coding strand. Okay, so this region is actually called what? Structural gene. The whole unit is actually called, this is structural gene. The whole unit is actually called what? Cistron. Can you understand? Cistron. And then replicon, where DNA is replicated. Unit of replication. Replicon is unit of replication. Okay, actually they are, you can study well in your higher education. Okay, can you understand? So, different regions of a gene. That's what is here. Molecular structure of a gene. A gene consists of different regions. Short lifespan, easy to cultivate, easily observable, 7 contrasting characters. Yeah. Short lifespan எழுதிட்டீங்களா? Okay, அதான் first short lifespan. Bisexual flower. It has bisexual flowers. Easily observable 7 contrasting characters. Hmm? You don't know that? Yeah, everything. Stem height. Please get your yellow book in page number 27. No, no, page number 27. See, look at that. Here. One table format is there. I too gave in your class. Same thing. Long time we are discussing that. Just check. Is it not there? Can you see? Can I see? Show me. So that they are the 7 contrasting characters. Here, get your yellow book. There you can see. Look at that. Seed color, seed shape. You want me to write? Don't worry. You finished up? Yeah, you can give it here. Give your book. 27. That table you please copy. Okay. Some questions for Vijay Lakshmi. Vijay Lakshmi, are you there?
Sir.
Yes. So, monohybrid cross.
Monohybrid.
Okay. Cross F2. In F2 generation, what is the phenotypic ratio?
Can you repeat the question? Can you repeat the question?
In monohybrid cross, in F2 generation, what phenotypic ratio it shows?
So 3:1.
Yes, correct. It is 3:1. Correct. What is the genotypic ratio? Same in monohybrid cross. Genotypic ratio. For example, TT1, TT heterozygous 2, and TT1. So your ratio is 1:2:1. That's the genotypic ratio. Can you understand, Vijay Lakshmi? Hello? Vijay Lakshmi. Fine. Can you understand that?
Yes,
Please
sir.
know that. Yeah. And then Kaushik, you tell me the phenotypic ratio of dihybrid cross. In F2. Phenotypic ratio. Tell me, Kaushik. Kaushik. What? But it is one important character, short lifespan. That's why he could able to cultivate and see the results in few weeks. நினைச்சு பாருங்க 6 மாசம் இந்த பிளான்ட் வளரும்னு சொன்னா அவரால எக்ஸ்பெரிமெண்ட்ஸ் பண்ண முடியுமா? பார்க்க முடியாது. few weeks, 2 to 3 to 4 weeks la adu results பார்த்திடலாம். Okay, Kaushik, are you there? Kaushik, say yes or no. I asked you a question. Okay, I think he's not there. Fine. You tell me, what is the dihybrid cross order F2 order phenotypic ratio? No, not genotypic I'm asking. It's phenotypic ratio I'm asking. 9:3:1. Correct. Okay, fine. Anyway, so since I have some work, so let me finish it a little early, 5 minutes early. And so please go through, students, well, and soon you will be having many number of tests, okay, in all of the portions. But first, be sure in genetics as well as means heredity, and then your reproduction in organisms. Okay, especially lot of questions will be asked from reproduction in plants. Since animal reproduction easily you can answer, so practice you need actually in plant reproduction. So use your study material, learning room study material, and please learn quickly. Thank you. Thank you, Vijay Lakshmi. What is? Let me check with sir. Okay, not going to start a new chapter, but kind of revision from the beginning, whatever like that only. Just environment is there. Apart from that, all the chapters finished. Just our environment. That we can discuss it later also. Okay.